right, here we go with organelles part 1. Okay, so the first, where my mouse? All right, here we go. First organelle we're going to talk about is the plasma membrane, and in our next chapter we'll talk a lot more about membranes and the plasma membrane. But what I want you to remember for this chapter is that the plasma membrane is found in all cells. Prokaryotic, eukaryotic, plant, animal, fungi, protists, all of those. And the purpose of the plasma membrane is to separate the material inside of the cell from the outside. The other purpose of the plasma membrane, and this is what we'll really start talking about next chapter, is to also regulate what goes in and out of the cell. So I just added a few micrographs. The plasma membrane is made up of a phospholipid bilayer, and we'll write that on the next slide. So just so you can kind of orient what you're seeing here, and proteins. And so this down here, this dark line, is showing a membrane, and it's actually pinching off. So what we'll talk about, kind of an interesting theme with membranes, is membranes are all made of the same thing, phospholipid bilayers and proteins. And so I like to think of it like bread dough, right? So like you make up your bread dough and you can pull a glob off and you can smush it back in, or maybe Play-Doh if you're not a baker. I actually don't bake, but I play around with bread dough sometimes, I guess. So they're all made of the same thing, so you can actually pinch off membranes like shown at the bottom here, and they can actually fuse back on also. So this dark line is showing you the membrane, this micrograph, so hopefully you recognize this. This is a transmission electron micrograph because it's giving you that flat view, black and white, but lots of detail. And what they're showing you is these are two cells next to each other, and they've zoomed in, I can't follow it very well, on the two plasma membranes side by side. So you can't really, we don't have, you can't see the phospholipid bilayer here. These are really two distinct plasma membranes, with each made up of a phospholipid bilayer. So let's write down that term, phospholipid bilayer. So it's right here. We'll talk a lot about phospholipids. Okay, that's this molecule here. Just kind of write down this term right now. Like I said, next chapter we'll get into it. And it's a bilayer because there's two layers. So hopefully you can see that this is one layer, these yellow heads and the tails, and this is another layer. And they come together to form a membrane. So all membranes are made up of a phospholipid bilayer. The other main component of a membrane are proteins. And we'll talk about the importance of these proteins, and these are all shown as these purple blobs here. They are what helps regulate and allow things to go in and out of the cell. So, plasma membrane, all cells, a phospholipid bilayer, and proteins. Something else you find in all cells is the cytoplasm. And you can see there's two very similar words here, cytoplasm and cytosol. And I really like this diagram. So let's first say that cytoplasm is in all cells. Okay. So prokaryotic cells, All this fluid is called the cytoplasm. And it's the fluid inside of the plasma membrane or the cell membrane. And it's where lots of chemical reactions happen. When we talk about cellular respiration, we'll talk about glycolysis. Glycolysis is this group of enzymes floating around in the cytoplasm that work together to break down sugars so that the cell can eventually use those sugars for energy. Cytosol is specific to eukaryotes. And it's still the same thing. It's still this fluid. The difference between cytoplasm and cytosol is that cytosol is basically the cytoplasm minus the organelles. Oops, I'm running my words in together. All cells. Right? So I think this diagram shows it nicely. So because this has a nucleus and membrane-bound organelles, this is a eukaryotic cell. So everything between the plasma membrane and the nucleus is called the cytoplasm, and that includes all these organelles. When you talk about the cytosol, you're saying it's all the fluid not including the organelles. So that's why I put cytoplasm minus organelles. I will admit, sorry, I gotta smudge up my glasses, that sometimes I get lazy and call everything cytoplasm, but the technically correct term for eukaryotic cells, not including the organelles, is cytosol. Okay, now we're going to talk about an organelle that is unique to eukaryotes. So remember that prokaryotes, oh, I probably just put my hand in front of the camera, sorry. Eukaryotes mean true nucleus, prokaryotes mean before the nucleus. Okay, so nuclei, which is the plural, are only found in eukaryotes. And the structure, so let's describe what you're looking at. The structure of the nucleus is it has a double membrane. And we call that the nuclear envelope. Okay. So it says nuclear envelope here, and hopefully you can see that there's 2 membranes. So there's 2 phospholipid bilayers right next to each other, the nuclear envelope. And then these proteins that cross those 2 membranes are called the nuclear pore, and they control what goes in and out of the nucleus. There is an area inside the nucleus. It does not have a membrane around it. It's just an area called the nucleolus. Hold on, I forgot to remove my keyboard. I don't have the room for my hands. Okay, where were we? So an area inside the nucleus equals nucleolus. So area, no membrane around it. And you've got what it says here, chromatin. So inside the nucleus, is where eukaryotic cells keep their DNA. So chromatin is technically DNA plus proteins. And the proteins help organize your DNA so it doesn't get all tangled up. Some very important functions happen in the nucleus. So in the nucleus is the site of DNA replication. That means copying your DNA. And we will talk about this towards about 3/4 into the semester. The other thing that happens in the nucleus is transcription. Transcription is when we copy a gene of DNA. Uh-oh. Once more this happens. There we go. We copy a portion of DNA called a gene to RNA. That's transcription. And then the other thing that happens in the nucleus, but more specifically in the nucleolus, okay, so in that sub-region area of the nucleus, is the assembly of ribosomal, and we'll talk about ribosomes in just a minute, ribosomal subunits. And subunits is a key word here, and I'll explain that in just a sec. Okay, so we have 3 processes that happen in the nucleus: DNA replication, transcription, the copying of DNA to RNA, and the assembly of ribosomal subunits. So here, just some images of a nucleus. So our little amoeba, we can see a dark nucleus here. We see this in our cheek cell that's stained. We see it beautifully as a green in this immunofluorescence image. And here, look how big this nucleus is. This nucleus is huge in this cell. And then this is the nucleolus region. So you really cannot see the nucleolus unless you're using an electron micrograph. So hopefully you recognize these as light microscopy or bright field. This is a stained cell, and this is a TEM, transmission electron microscopy image. So that's what your cell looks like. That's what your nucleus and nucleolus look like. Okay. Nucleus versus nucleoid region. So prokaryotes have a nucleoid region. They don't have their DNA surrounded in a membrane. So the nucleoid region is where DNA hangs out in prokaryotes. And one thing I want to point out is there's a little bit of difference between prokaryotic and eukaryotic DNA. Now, structurally, what they're made of is the same, but in general, eukaryotes have linear chromosomes. So you may or may not know that you have 46 chromosomes. Right? So we sometimes draw them like these little fat guys, and you have 23 pairs of them. I'm not going to draw them all. Sometimes you'll see them look like these X's. That's after they've replicated, but notice they're linear. They have a start and a finish. Prokaryotes, and I should say eukaryotes, have many linear chromosomes. So again, humans, 46 chromosomes. Prokaryotic cells usually have one circular chromosome. hard time writing. It's not, oh. I'll get better. Okay. So, remember that all cells have DNA, and now we're talking about some differences. Prokaryotes have circular DNA, usually only one chromosome. Eukaryotes have linear DNA, usually multiple chromosomes. And your book talks about chromosome and chromatin. And so let me help you define this. So chromatin versus chromosome. So they're both DNA plus proteins. And you may notice in your guided notes, I put a lot of compare and contrast. I'll give you a hint. On your post-class quizzes, you'll probably have a compare and contrast question. Okay, I like to have you talk about similarities and talk about differences between, say, parts of a cell or different processes that we discuss. So, hint, hint. Chromatin just means that the DNA and proteins that help organize it are kind of loose. So they're not like a chromosome, which is compact. So chromatin is loose DNA. Excuse me. Chromosomes are condensed. I should put DNA plus proteins because they're both DNA plus proteins. So when we talk towards the end of the semester about mitosis and meiosis, we'll talk about how early on the DNA is just all these linear DNA pieces are just all stringy there, and then as we get ready to divide the cell, they all get nice and condensed and organized. Okay. The last organelle we're going to talk about in this lecture are ribosomes. Ribosomes are found in all cells. So this organelle lecture is really about what's in all cells, right? We have DNA, we have cytoplasm, we have ribosomes, and we have a plasma membrane. The function of a ribosome is to make proteins. And this process is called translation. Right over here. So the process of making proteins is called translation. That's what ribosomes do. All cells, ribosomes in prokaryotic cells, ribosomes in eukaryotic cells. And the structure of them are slightly different. But the function is the same. So let's look at what a ribosome is made of. And ribosomes are made of proteins and something called rRNA, and the R stands for ribosomal RNA. Remember that in eukaryotes, the ribosomal subunit, so the protein plus the rRNA, is assembled in, that's right, subunit, assembled in the nucleolus. Okay. And look, this is what I mean by a subunit. Ribosomes have large and small subunits. So the whole ribosome doesn't come together until it's in the cytoplasm. So the ribosome, which is the small and large subunit, is assembled in the cytoplasm, or cytosol. See, I told you I'd get lazy. We'll do this cytosol. For prokaryotes, because they don't have a nucleus and they don't have a nucleolus, the proteins and ribosomal RNA are just put together in the cytoplasm of a prokaryote. But for eukaryotes, the large subunit is made in the nucleolus, and then it's exported out of the nuclear pore, and the small subunit is made in the nucleolus and exported out of the nuclear pore, and then when it's time for translation, the two subunits come together. And so this electron micrograph, all these little black dots over here are showing ribosomes, and this is actually on the rough ER, which we'll talk about in the next lecture. Okay, I think that is good for organelles part 1. I put it in a thing. So next we're going to really focus on the eukaryotic organelles.